Assalamu alaikum students. Today we are going to tell you about the factors affecting the rate of enzyme action. Enzymes are very sensitive to the environment in which they work. So any change in the chemistry or a shape of enzyme molecule can affect its activity. There are some factors, for example, temperature, substrate concentration and pH. First, we'll tell you about the temperature, that how temperature is going to affect the enzyme activity. When we increase the temperature, then with the increase of the temperature, the enzyme activity is also accelerated. They also get increased. But there is a limit of that temperature according to the different bodies, according to the different organisms. For example, in human body, the maximum, or you can say the optimum temperature in which the enzymes work at its best is 37 degrees Celsius. So if the temperature increases beyond that limit, beyond that optimum temperature, if it crosses 37 degrees Celsius and increase like 39 or 40 degrees Celsius, then what happens? The enzyme activity starts to decrease. And why it starts to decrease? Because there is the denaturation of enzymes. The globular structure of the enzyme is lost, and because of that, the enzymes get denatured. You can see the graph as well, where the increase in the temperature increases the enzyme activity, but there is an optimum temperature, and we are showing you that this is the maximum temperature where the enzyme activity will be maximum at its maximum. And when that temperature will be going on increasing, then what will happen? There will be a decline in the enzyme activity. Okay, the next factor is substrate concentration. If enzyme molecules are available in a reaction, so increase in the substrate concentration increases the rate of the reaction. If enzyme concentration is kept constant and amount of the substrate is increased, then a point is reached where any further increase in a substrate doesn't increase the rate of reaction. Why is it so? Because there are no more enzyme molecules, because there are no more active sites which are available, which has to be available for the substrate to bind, for them to make an enzyme substrate complex and to go on increasing the rate of a reaction. So there will be a constant, you can say that the rate of reaction will be constant, and there will be a state reached which is called as the saturation state, which means no free active sites available for more substrate. And you can also see the graph of this effect of the substrate concentration on enzyme activity. The next factor is pH. Now, what is actually pH? The pH is the potential of hydrogen, and all enzymes work at their maximum rate at a narrow range of pH, which is called as the optimum pH. A slight change in this pH causes retardation in enzyme activity or blocks it completely. Every enzyme has its specific optimum pH value. For example, there are some enzymes which are present in the small intestine, and here in the small intestine the medium is alkaline, so there is the trypsin enzyme which is used for the breakdown of the protein molecules. On the other hand, inside the stomach, where the medium is environment, you can say that is acidic medium, and here the enzyme which is used for the breakdown of the proteins is pepsin. So enzymes vary according to their environment, according to their medium where they are present. So according to that, they are performing at their maximum. So if, for example, if we say that pepsin enzyme which is present in inside the stomach and the medium is alkaline, then what will happen? The pepsin will be not performing according to its like it has to be performing. So it will not be breaking down any further protein molecules. So it will be the proteins will be present as it is. and there will be no change in the protein molecules. Similarly, if the trypsin molecule, the trypsin enzyme, which is present inside the small intestine, and here the medium which is present, alkaline medium, instead of that we change its medium into the acidic medium, then what will happen? The trypsin is not going to react on the proteins molecule, so the proteins will not be broken down here. So change of the medium, that will also affect the working of the enzymes. And you can see the graph of the pH too. Now coming to the assignment part, there are different short questions and a long as well. What is optimum temperature? What is optimum pH? Define saturation and substrate concentration. And this is the long question. Explain factors affecting the rate of enzyme action, in which all of the factors you have to explain them and you have to draw the graph as well. Thank you.